Dear Senior 2 students, you are most welcome in our fifth lesson in a topic, transport of materials. And in this case, we are going to talk about transport of manufactured food or products of photosynthesis in the phloem tissue of the plant. So we expect to talk about the structure of phloem, the products, how they are transported, and the evidence of this transportation. To start right away, let's start with the phloem tissue, a tissue responsible for transportation of manufactured food or products of photosynthesis. This tissue is a compound tissue with two types of cells, the sieve tube element and a companion cell. The sieve tube elements, they lack components of a cell. Therefore, they are supported by another cell called a companion cell. Just the word dictates a companion to give support or company to the other cell with few living components. The companion cell, it's a living cell to give support to the sieve tube. Then within the sieve tube, there is this perforated opalous structure called the sieve plate to allow the flow of material from one cell to the next cell. Then it has the thick wall, it's hollow, they are long tube and cylindrical to allow transportation of manufactured food. Adaptations of this phloem tissue, they have living companion cells to give support to the sieve tube. Then they have elongated whole sieve tube to provide a space or a tube for transportation of manufactured food. Perforated with cross wall, a sieve plate to allow movement of manufactured food from one cell to the next phloem cell. Then they have numerous mitochondria in the companion cell for the active loading or pumping of manufactured food into the sieve tubes. Then we have a guiding question here. How is the phloem tissue adapted to its function? Please attempt this question. Then how is the translocation of manufactured food occurs? The products of photosynthesis are made in the region, which in this case we are going to call the source. For example, the plant leaves are manufacturing food, therefore they qualify to be called the source of food. Then you have another region called the sink, where the manufactured food is either stored or broken down in respiration to release energy. So in this case, at the source, food is manufactured. So there is a lot of food manufactured from the leaf, and it is packed into the phloem, the adjacent phloem tissue. Then at the sink, a lot of food is either used or stored in this form. Therefore, at the source, the phloem tissue, due to loading of food from the source, is going to absorb water from the surrounding xylem tissue and then cause a bulk transport of food from the source to the sink by means of mass flow. Water, food, then packed in water and transported as a bulk. That is transportation by mass flow from the source to the sink. We say that the source food is manufactured, and at the sink food is utilized. Then do we have evidence to demonstrate this? Yes. We have mainly three evidence here: the ring experiment, the feeding aphids, and radioactive tracer. Under ring experiment, if you cut the bark of the tree into a circle as shown here in the drawings, then after some time, like half a year, you are going to see the upper part of the stem becoming swollen. And this implies that you cutting off the bark interrupted the movement of food from the source, from the leaves down to the roots. So food kept on accumulating on the upper part, causing it to swell. So that is a ring experiment. Then we have a feeding aphid. An aphid, as it is feeding here, demonstrated on the drawing, it pierces its stylet deep into the phloem tissue. So if you cut off the aphid and remain with the stylet, here the stylet is going to release a liquid, and if you test the content of this liquid, you are going to find manufactured food. So that is how the feeding aphid is used to demonstrate the process of translocation. Then radioactive tracer, here this is like we use carbon, you use labeled carbon dioxide. For example, use carbon 14 to label it, and it is used to make carbon dioxide, which you supply for photosynthesis, and at the end you find food which has been manufactured using that labeled in quotes, labeled carbon dioxide in the phloem tissue. It implies that manufactured food is translocated in the phloem. For more information, please check our e-learning platform to get the notes, and then you compare with this explanation to get the detailed overview of this concept. Otherwise, I wish you the best. Please stay safe. Thank you.